So now we're in the lymph node. We're looking at how HIV, brought to the lymph node by either one of our well-intentioned dendritic cells or maybe by a macrophage, manages to infect our T helper cells, aka our CD4 lymphocytes. Well, it all sort of comes down to receptors. On our T helper cell membranes, we have 2 to 3 receptors that help HIV gain access to these cells. We have the CD4 receptor, which is sort of the primary receptor that HIV needs, and we have either one or both of CCR5 and CXCR4. CXCR4 being much less prevalent. CCR5 is really the preferred co-receptor that most strains of HIV need to bind. But these are both called co-receptors in this context here. So what's important about these particular receptors is that HIV happens to have a protein, this thing here called GP120, on its envelope, the outer part of the virus here, that interacts really well with these receptors. You can almost think of it as a lock and key setup. These receptors here are some of the locks that need to be opened to gain access to our T helper cell. And unfortunately for us, HIV has the key, this GP120 protein on its surface. So how does it work? Well, GP120 on the HIV envelope, it first binds the CD4 receptor on a T helper cell. This binding between the two sort of induces a conformational change in the CD4 receptor protein here, and it allows our co-receptor here, either CCR5 or CXCR4, to grab a hold of this complex and pull the viral membrane and the T cell membrane closer together. And when they get close enough, this little stalk here, the stalk of the GP120 protein, it sort of pierces through our T cell membrane, and it pulls the viral and the T cell membranes even closer together. And what ultimately happens is that the two membranes will fuse together, which allows the HIV particle to essentially inject. Now that it has access to the inside of our T cell here, it injects its genetic material into our T cell, inside this viral capsid here. And that's in the form of viral RNA, ribonucleic acid. And this envelope is just sort of left at the surface of our T cell here. So once this capsid with viral RNA and some viral enzymes enters our cell, it gets degraded by some of our cellular enzymes. And this is essentially another bad move on our cell's part, because it releases the viral enzymes and the viral RNA, which can now get to work on taking over our T cell. We kind of just got Trojan horsed in a way. And you know, most viruses at this point are happy to just use our cellular machinery, like our ribosomes and our cytoplasmic nucleotides, to make copies of themselves, and they leave our DNA alone. But not HIV. HIV is a little sneakier than normal viruses, and probably the key viral enzyme among all of these that allows it to be so sneaky is this reverse transcriptase enzyme. So what reverse transcriptase does is it takes this viral RNA here, the one that came with, and it uses some of our nucleotides that are floating around in the cytoplasm to revert this viral RNA into a single strand of DNA. It uses the viral RNA as a template to synthesize a strand of DNA. And then it synthesizes a complementary strand of DNA for the single strand. So we end up with some double-stranded viral DNA here. And so right now, alarm bells are probably rightfully ringing in your head, right? We just went from single-stranded RNA to single-stranded DNA to double-stranded DNA. That's not supposed to happen. Remember the central dogma from your biology classes that said we go from DNA to RNA to proteins? And here we are doing the exact opposite of that? Well, that's exactly what's happening. We are doing the opposite, and that's why HIV is called a retrovirus. It subverts the central dogma and it generates viral DNA from viral RNA. Outrageous. The other sneaky thing about reverse transcriptase I wanted to mention is that it makes a lot of errors doing all this polymerization, all this synthesis. It doesn't quite have the proofreading ability that our DNA polymerase enzymes have, that obviously come in handy when we're replicating our DNA. So on a practical level, what this means is that, well, A, lots of viral polymerase errors equals lots of mutations in the viral DNA. And that means that over time, the virus can develop resistance to certain antiviral medications because the medication eventually might not be able to even recognize the viral DNA. And B, it's really hard to make a vaccine against HIV because even small changes in the genes of the virus might render the vaccine ineffective. Remember, we're talking about one cell here and the mutations that happen within one cell, but keep in mind that there's potentially going to be millions of HIV-infected T cells in which HIV will be mutating ever so often. And actually, this reverse transcriptase enzyme is one of the targets for some of the medication we use to control HIV levels. We try to block this viral reverse transcriptase enzyme from working. Anyways, back to our now double-stranded DNA here. Another viral enzyme called integrase will come along and grab hold of it. It'll then bring it to the T cell's nucleus and carry it through one of our nuclear pores into the nucleus. From there, and this is sort of the point of no return in an HIV infection, the integrase enzyme nicks, it makes a little cut in our human T cell DNA, and it allows this double-stranded HIV DNA to integrate itself into our DNA. And this step essentially establishes the lifelong infection with HIV. The viral DNA has sort of now become congruent with our own DNA. So from here, a few different paths can be taken. Well, either this DNA just sits here and isn't actively transcribed into mRNA, and we call this a latent HIV infection, when your cell has integrated viral DNA into your own, but is not actively doing anything with that DNA. Or your DNA transcription enzymes might come along. This is usually the more likely thing that happens. So let's say RNA polymerases come along. Well, it's going to transcribe this viral DNA here just as if it was your own DNA. So it'll start cranking out viral mRNA transcripts, which then leave the nucleus. They find some ribosomes, say on the rough endoplasmic reticulum, and they start to use the ribosomes to make proteins, like new envelope proteins, for example. These envelope proteins will then make their way through our endoplasmic reticulum, head up toward the cell surface, right, our cell membrane, and once enough get up there, they start to coalesce a bit, they cluster together. And this is actually happening in a lot of other places on our cell membrane. See, you can see all the new GP120 proteins here on the surface of these viral envelope segments. And while this is happening, another key viral protein is being made at the same time. Actually, it's a viral polyprotein. Poly because it's essentially multiple different viral proteins laid out end to end on one long protein strand. So these will include those viral enzymes we talked about earlier, right? Reverse transcriptase, integrase, as well as some other proteins that the virus needs to be infectious. So all of these long viral polyproteins, along with some viral RNA, they also get brought up to the surface, to the areas where all the envelope proteins have clustered together. And so now, all of these components can come together to start forming a new immature HIV particle. And I say immature because it's not quite ready to infect other cells yet. One more thing has to happen before it matures and it's ready to be infectious. It needs help from yet another viral enzyme called a protease. Proteases are special enzymes that cleave up polyproteins, just like this one here, into smaller proteins. But they only cut at specifically marked sites. And this protease here, it does just that with this long viral polyprotein that's been made using our ribosomes. So it snips it at a few different sites, and we end up with what are all the components that an HIV particle needs to infect. So for example, this might be its reverse transcriptase here, and this might be its integrase here, and so on. So the protease starts cutting things up, and while it's doing its slicing and dicing, these components here together, they all start to bud off the T cell as a new virion. And shortly after it buds off, the protease is finished cleaving this long protein up. So this is now a fully mature HIV particle. Now that it's used this T cell to be made, it's ready to go on and infect other cells, particularly, of course, T helper cells. But what happens with this T helper cell that was infected? Well, our old understanding of it was just that as tons and tons and tons of new virions budded off of our infected T cell, all of that budding off would actually kill our T cell. But recently it's been discovered that things are a lot more complicated than that. In the vast majority of cases, the T helper cell does die, but it's not because of the budding off of the virions. It's most often because infection and subsequent production of HIV particles, it sort of triggers this sort of self-destruct mechanism within the T cell. But I'll cover that in more detail when we talk about how HIV kills our T cells.